Alexander Hoffmann. He is the director of the Institute for Quantitative and Computational Bioscience and the Thomas Escher Professor of Microbiology in the Department of Microbiology, Immunology, and Molecular Genetics at the University of California, Los Angeles. His research interest is the development of a predictive understanding of how cellular and the molecular networks regulate immune response. I have also think here to read about Alex, but I think that it will be better to let him speak. Alex, thank you very much for accepting our invitation. We are here. We are ready to hear you.
Oh, well, thank you so much for the invitation. I'm very pleased to be able to be here. And so greetings from Los Angeles to Brazil. I have my shirt on, which is the QC Bio Institute, Quantitative and Computational Biosciences. And some greetings to my colleagues and the students. And I'm excited to share some of our research. And let me share my screen. And which I think, is that good?
That's perfect.
Great. So the title of my talk today is a temporal signaling code to specify immune responses. And this is research that actually began a long time ago in my laboratory, but continues to be exciting because it continues to provide new insights. So let me introduce what we think about immune responses. Immune responses begin, of course, when pathogens like viruses infect tissue cells. And within those tissues, there are the tissue cells like hepatocytes or fibroblasts, but there are also macrophages. And macrophages are present in every tissue. And their primary function is to sense pathogens and then initiate an immune response. In fact, macrophages are the first responders or first sentinel cells that sense pathogens, and they can defend against the infection by eliminating the pathogen directly through their phagocytosis activities. But if that is incomplete or insufficient, then they can cause an immune response through inflammation and other immune activation. And one of the first things that, of course, has to happen is that neutrophils or other specialized immune cells will come to the site of infection and unload their toxic cargo to eliminate the pathogen. And then also dendritic cells will come and take pieces of pathogen and present those pieces of pathogen to the adaptive immune response. And that generates the adaptive immune response with antibodies and cytotoxic T cells and so forth. So macrophages play a really key role in this, and they have to decide what type of immune response to activate and when, because this happens in minutes, hours, or days. So in fact, macrophages have to distinguish different types of pathogens. There's viruses, there's bacteria, there's eukaryotic pathogens like fungi, And also there's tissue damage that's not really about pathogens, but it still needs an immune response to repair the damage. And even within the big class of viruses, think about that some viruses have genetic material that's DNA, some have genetic material that's RNA, single-stranded or double-stranded, and some are inserting their genetic material into their host cells, and it becomes part of the genome. So there are very different kinds of biology for each of these pathogens, and each pathogen, therefore, requires a different response, a response that is specific to the pathogen to be effective. So, for example, cell intrinsic defenses can involve strengthening cells or leading to apoptosis of cells, depending on what the pathogen is. It requires different types of local immune responses where different types of immune cells are recruited to the site of infection, and also requires different types of systemic immune activation with fever and inflammatory and anti-inflammatory mechanisms. So the macrophage actually has to provide an appropriate response for any of these types of pathogens. And so people have been studying this, of course, for years. And so we describe this the specificity really as a response specificity that is a hallmark of healthy macrophages. And this response specificity is further modulated by the tissue context and also actually by the prior exposure of macrophages to previous pathogens. So actually macrophages have a memory as well. And that memory informs how they will respond in the future. So macrophages are really quite complex. And if this response specificity is not happening correctly, then in fact, this is the hallmark of many types of diseases. And of course, allergic responses, but also things like Alzheimer's and lupus and celiac disease and Parkinson's and stroke are all macrophage misregulation contributes to all of these. So that's why we are really keen to understand macrophage functions. And people have been since even in 2004, with a pioneer of systems biology, Kitano, summarized the literature and showed that there's so many molecules, this molecular network in the macrophage is very complex. And here are the receptors that sense pathogens, the pathogen sensors in the plasma membrane. And there's other pathogen sensors in the cytosol. And they're connected to signaling pathways. And the signaling pathways activate transcription factors. But what this network doesn't really tell us is how the stimulus response specificity works, because if you connect the lines from each of these receptors, they all go to the same transcription factors. So this network does not provide an answer to the question of how do macrophages generate pathogen-specific responses. And so one aspect that this map does not describe is the fact that signaling is dynamic. And this is where this early experiment I did when I was still a postdoc showed that NFκB, for example, a transcription factor that's activated here in minutes by immune stimulus like TNF, is activated, but then is also inactivated and reactivated, inactivated and reactivated. You can see there's a dynamic in the transcription factor activation. And Mike White's lab showed a couple of years later that this is also true in single cells using fluorescent reporters, where NFκB, the transcription factor NFκB, is fused to a fluorescent protein. And so this led to the hypothesis that actually the signaling pathways that activate NFκB contain a temporal code where the receptors are not on-off switches for each pathway, but they are more like telephone transducers that listen to the outside of the cell and then produce a signal that goes all the way to the signaling pathway to the nucleus. And thereby they can communicate to the nucleus what is on the outside of the cell. And the way they do that is through the temporal modulation of kinase activities or the temporal modulation of transcription factor activities. So this hypothesis was very exciting about 20 years ago when we first posed it. But when we say there's a temporal code, what we really need to address is what are actually the units of information? Because when I have a code, I can know that there's a genetic code, and that contains 3 nucleotides that are a codon. Or I have a Morse code where I can tap, something like this on, SOS, we know what the Morse code is. So if I say that is a temporal code, we have to define what the codons are. What are the packets of information that are meaningful? And so that is the question that we started addressing a few years ago and asked, what are the codons, which are the packets of information within this temporal code? How are they encoded by the network that are close to the receptor? And how are they decoded by the genes that are in the nucleus? And so I'll give you a short summary of a series of publications that we had on this over the last few years. The whole story began when Brooks Taylor, who was a bioengineering doctoral student with me, very talented, developed a image analysis pipeline that allowed us to image many cells in a video microscopy and extract the quantitative information of NFkB signaling from it. And so we produced a mouse in which NFkB, the transcription factor NFkB, has the fluorescent protein M. venus fused to it. And therefore, we can generate primary macrophages, healthy macrophages that are not immortalized, that come straight from the animal. And we're able to stimulate them with pathogens or pathogen-derived substances and obtain signaling dynamic information. And this project was continued and completed by Ade Adelaya. And here's the type of data that we obtained. We see, for example, here that there are many macrophages. If I just look at this one here, you see that NFkB in the nucleus goes into the nucleus and back out of the nucleus. And when that's quantified, you see the traces here. And here's another cell. So in response to this cytokine TNF, NFkB clearly is oscillatory. We also see that the 2 cells are not in synchrony. They are asynchronous. There's heterogeneity. And in response to LPS, we see that NFkB is in the nucleus, and it pretty much stays there. It is non-oscillatory. And when we look at the traces, again, we see some little variation here and maybe some degree of ups and downs, but it's nowhere as close as the oscillatory pattern on the left. So the same types of cells stimulated with different substances lead to different dynamics of NFkB. Now, of course, in order to account for this heterogeneity, we have to do statistics. And in order to do statistics, we have to generate hundreds of data points for hundreds of cells, not just 2. And so the automatic image analysis pipeline was important for this. And so we could summarize the data in this form, where this is a time course you see from 0 to 12 hours. And you see NFkB in the nucleus is in red. That's the red color. And here are cells. There are about 300 cells. And first, NFkB goes into the nucleus, it comes back out, it goes in, it comes back out, and so forth. It continues to oscillate. Almost every cell is oscillating. And on the right is another batch of cells. They're stimulated with LPS, and you see they're non-oscillatory. Later, they begin to see some oscillatory pattern. Also notice if I take the average of this, that's down here, this does not look oscillatory. So that's because these cells are asynchronous. So the average behavior does not show what is actually going on. We have to have single cell data. So, so the question is, we can see now there are oscillations and no oscillations, but maybe there's some other dynamic features. What are the other dynamic features that are different between the two? And if they're different between the two, they are informative of the stimulus. They carry information. And so we generated a lot of data, not just for one dose of TNF, but 5 doses of TNF, 5 doses of PAMP-CSK, which is bacterial ligand, and 5 doses of CpG and LPS, many doses of LPS, and gram-negative bacterial cell wall component, poly-IC, which mimics viral DNA, viral RNA, and we obtain data sets for each one. And so now we can ask which dynamic feature carries information about the stimulus. And to do that, we decomposed these complex dynamics into many different types of features. Of course, we have time series data, which are individual data points at each time point. But at each time point, we can either read off the amplitude, or we can read off the integral, or we can read off the derivative. Each time point is associated with many different types of things, so 6 or 3 different amplitude, integral, or derivative. We can also ask more complex questions like what is the peak activation? Or what is the duration above a certain threshold? Or what is the Fourier transform? What are the frequencies? And what are the primary frequencies and secondary frequencies? So we generated about 918 of these potential features about the data, decomposed the data into these 918 features, and then asked which of these features provides information. And to do that, we used Shannon's information algorithm, which of course was developed at a time when the telecommunications industry was laying big cables under the Atlantic and the Pacific, and it was important to measure the information transmission capacity of these new cables that went for long distances. And so, so the information algorithm or mutual information is essentially a statistical test that relates the input to the output. And in this case, the input is the stimulus and the output is what are the features of NFkB that the nuclear target genes are seeing. And what we found is that there are 6 dynamical features of those 918 that are sufficient. It's the speed of activation, whether the cells are oscillating or not, how long the duration is, what the peak amplitude is of the signal, what the total activity is, and how much of the total activity is early versus late. And those 6 codons provide all the information in that data set. And so they are a way of decomposing complex dynamical trajectories into just 6 dimensions. And you can see that here, that, for example, the speed codon or speed is in response to TNF is pretty high, whereas in response to poly-IC, it's pretty low. You can see a distribution because every cell is slightly different, but here you see that the distribution is centered around a high value or a low value. Or oscillations are very high for TNF, but low for LPS, as I showed you. Or total activity varies, and so forth. And so signaling codons are deployed in a stimulus-specific manner. To show you that these comprise all the information, it's not just information theories. Also, we can use a machine learning classifier, like an ensemble of decision trees, to take the trajectories, either using 918 features or just the 6 codons, and train the machine learning algorithm on these features with 5 different stimuli. And we can determine the F1 score, and we see here that the F1 score hovers around 75 or 80% in most cases. And we see that really the 6 signaling codons capture the full precision of all 119 also. So thereby, we focused on these 6 signaling codons. But the machine learning algorithm also can tell us where there are sources of confusion. We can see here that the F1 score or the precision is between 70 and 80%. But we also see a source of confusion. And the confusion here particularly is between PAM3CSK and LPS. Now, both of these are bacterial ligands. So maybe it's not that important for the cell to really distinguish between the two, right? Because they're both bacteria. But the cell does a very good job in distinguishing between poly-IC and TNF, for example. There's almost no confusion, right? So do these signaling dynamics really matter? I showed you this all in macrophages that are healthy. How about macrophages that are not healthy? So these come from an animal model of Sjögren's syndrome, which is an inflammatory syndrome. And this inflammatory syndrome is characterized by spontaneous episodes of inflammatory pathologies. So we used an animal model for this disease, bred it into our NFkB reporter, obtained macrophages, and imaged them, and used, as I showed you before. And then decomposed the data, and we used TNF, LPS, and poly-IC, and decomposed the data into our 6 signaling codons. And we find that, each signaling codon has a very specific pattern for these 3 different stimuli. And we find that the healthy or Sjögren's syndrome macrophages really show no difference in the speed codon. They show no difference in the peak amplitude. But they do show a difference in the oscillations, where TNF and poly-IC show nice oscillations, but not in the Sjögren's syndrome. Total activity is perfectly fine. The duration is affected, but early versus late is not affected either. So 2 signaling codons are affected, but the other 4 are not. Now, does that matter? Well, if we train this machine learning classifier on the data, in this case, the healthy data shows about 80 to 90% precision in this particular data set. And that's great. In the Sjögren's syndrome macrophages, there is precision in recognizing LPS, but not in TNF and poly-IC. In fact, there is big confusion. When the true ligand is TNF, the cells are responding as if they were responding to LPS. So what that means is the cells are not recognizing when their neighbor is giving them a TNF signal, but they're interpreting that neighbor signal as if they're getting infected by a bacterium directly. They're overreacting. Right? So Sjogren's syndrome, therefore, in this model looks like a inflammatory confusion disease. The cells are reacting by confusing the stimuli. So the signaling codons matter. They're important. So now let me ask the question of how are they encoded? Well, there are receptors, as we know. There are receptors, and the receptors activate the kinase IKK, and the kinase IKK leads to the degradation of the inhibitor protein IKB alpha. And when the inhibitor protein IKB alpha is degraded, NFkB can be activated. But then, of course, there is the well-known negative feedback loop where NFkB is responsible for activating IKB alpha. And so that is what's setting up the oscillations. And so how is this possible? We tested this model, again, with an IKB alpha knockout. And indeed, whereas wild-type cells oscillate in response to TNF, the mutant cells do not oscillate in response to TNF. So the negative feedback is important for the oscillations. But how can the same model also generate non-oscillations? So for this, we did Hopf bifurcation analysis. Hopf bifurcation analysis, it describes how the steady state is a function of how much IKK there is. And when we see that this indicates a divergence of two steady states, this is where the cells are able to oscillate in. This is in essentially the regime 2, which is where at NFkB is able to oscillate. Even at a regime 3, when IKK is a little bit higher, the cells will still oscillate. You see medium levels of IKK oscillations, slightly lower levels of IKK oscillations. But when the cells have a very high level of IKK, interestingly, the oscillations go away. High level of IKK, no oscillations. So now we can see that actually whether the cells oscillate or not depends on just the amount of IKK. The amount of IKK. And interestingly, the amount of IKK does not really affect the period of the oscillations. It's almost a flat line. The period is hardwired into the network. But whether the system oscillates or not depends on how much IKK there is. So with this insight, I think it's that this schema is essentially true, that IKK activates either NFkB that is oscillating or not, and this is happening in the core module. And so what the rest of the network has to do is to determine how much IKK there is and at what time. And then the other 5 signaling codons are all determined by these other signaling networks that are associated with each receptor-specific modules. And so I can blow this up a little bit. Here we have on this picture IKK and the negative feedback loop I described. And IKK is regulated by another kinase TAC1. TAC1 is a function of TRAF6, which is a function of MYD88 and TRIF, or TRAF2 in this case. And so these signals, so for example, from the TNF receptor up here, TNF receptor is trimerized, activates TAC1, which activates IKK. Or a little bit more complex, we have LPS activating, dimerizing the receptor, leading to MYD88 activation, leading to TRAF6 and TAC1 and IKK. Or LPS is also able to go to the endosome, and in the endosome activates TRIF, and TRIF activates, endosomal receptor activates TRIF and activates TAC1 and IKK. So this is essentially the network. And so now what we find is actually through work that was just published in Nature Communications by Chalu Guo, a really talented postdoc in my group, is that these simulations are capable of recapitulating all of the experimental data. So the low dose of TNF, this is the experimental data. These are the simulations. Experimental data for medium dose, simulations. Experimental data for high dose, simulations. And you can see it captures the dose responsiveness, it captures the speed, the amplitudes, the other features, and the heterogeneity as well. And for CPG, things look completely different. Again, the simulations match the experiments. And for PAM3CSK also, and for LPS as well, and for poly-IC. So we have a model actually that is remarkably good given how simple the simple principles of signaling networks that we have in this biophysical model. And so I can say we now understand how the signaling codons are activated. And the data we have for this comes from population from measuring single cell responses in a population assay, essentially, right? Where every dish, 1 million cells or so, are being stimulated with TNF, and we get a response, or PAM3CSK, we get a response. And this is how we've calculated the stimulus response specificity. But what we have not been able to do experimentally is to determine how each cell is responding differently. We only know that the populations of cells are responding differently. But each cell may respond differently, but we cannot do that experimentally because we can't stimulate the same cell multiple times, because when we've stimulated a second time, it still remembers the stimulus of the first time. It's a classic Heisenberg uncertainty principle, right? Where things are affected. So, but computationally, we can do that. We can take cells, our individual cells that have been parameterized by the mathematical modeling, and these virtual cells we can stimulate with each of the 5 ligands, and we find that maybe some cells respond very differently to the 5 ligands, sort of like this, and some cells may not respond differently. They respond, but they all look the same. This is just hypothesis. And so you can see that actually for each cell, we may determine whether it's specific or it's not specific. And that's what we did. And here are now individual cells that are simulated in response to TNF, PAM, CPG, LPS, and poly-IC. And you see there's different responses. And then we can measure how many pairwise distinctions there are and whether cells are able to distinguish each pair, TNF from PAM, PAM from CPG, TNF from CPG, and so forth. There are 10 possible pairs, and about 1/3 of the cells do it perfectly for all 10 pairs. Another third do it well for 9 pairs, but 1 is not good. And then the remaining third are distributed. But we also see that there are no cells that are really, really bad. So that's good. And interestingly, when we look at where the confusion is for the cells, here are the 10 pairs, and here on the y-axis are all the single cells. We see actually that the confusion occurs in different pairs. So that means the confusion occurs, in different pairs. Every cell has a different problem. They don't have the same problems. They don't have the same flaws. They have different flaws. And so that means the cells differ actually in their blind spots. Or in other words, there is a diversity of flaws, which is actually kind of like we as humans are, right? We don't all have the same problems. Or the same flaws. We have different flaws. And one of the results of that is that consortia of cells, or small groups of cells, like groups of 2 or 3 or 4, actually do really well in distinguishing all stimuli if they work together, right? And so with 4, even 3 cells, they almost have 100% efficiency in distinguishing all 10 pairs. Whereas if the flaws were all correlated, they were not diverse, they were all correlated, the same distribution of flaws were correlated, this would not be working so well. So the diversity of flaws allow consortia of cells to make better decisions, which is a really nice conclusion, I think, because it relates to also how groups of people can make better decisions because we complement each other for our blind spots. But let me go back to the question. I think I showed you what our signaling codons are. I also showed you how they're encoded. And now let me say something about how they decoded. Well, how do target genes of NFkB distinguish between different signaling codons? And we have 6 of these. So actually, we looked at each one by one. And so, for example, the level of detail we wanted to get into is to generate data sets that are not just about the RNA, the messenger RNA of poly-A plus mature messenger RNA, but also chromatin-associated RNA that contains introns, that is closer to initiation. And these are all time course data, 0 to 8 hours, 0 to 8 hours, right? In response to different stimuli. And we're using healthy cells or mutant cells. And so these data sets then are used to describe whether particular mathematical models of gene expression, of gene regulation, are consistent with the data. And if they are consistent, we provide a yellow indicator. Here are all the genes we're considering, about 80 or so genes in this particular study. And so we see that some models don't do very well. Other models do better, but we also see that some genes don't even match the best models. So we still have more work to do to really understand every gene. But for many genes, we have a good understanding how well they work. And so, for example, the duration of short duration or long duration NFkB is decoded by 2 mechanisms. One is a slow chromatin step, and the other one is long mRNA half-life. And the 2 come together to generate different levels of RNA that are stimulus specific. And for the 80 genes, we can find that these genes primarily depend, their specificity primarily depends on mRNA, long mRNA half-life. And for these genes, their specificity depends on the slow chromatin step. And for these genes, it's a combination. And you also see that for some genes, the gray bar actually indicates they are highly specific, but our model does not capture that specificity. So there's other mechanisms in addition to these 2 that are also important. So with this, mathematical modeling doesn't lie. It shows the truth. It shows that we can understand some things, we understand some things, but other things we do not understand yet. And this is described in this paper in Cell Systems. So to some extent, I think we can say that we are beginning to learn how duration is decoded. And we also had studies on the speed, and we had studies on the amplitude and so forth. But one of the most interesting ones, of course, is oscillations and non-oscillations. And that continued to be a big question because years ago, we actually published a paper that showed that some oscillating cells or some non-oscillating cells actually activate genes exactly the same way. There's no difference. So this was published as negative results in Science in 2005. And why was it published in Science? Because everybody expected that oscillations are important. And this data clearly shows they make no difference. But still, we still didn't have an answer. Even after 15 years later, there was no answer to this question of oscillations. Until we realized that maybe we shouldn't be only looking at immune genes that are poised, that are on where the DNA is available for NFkB to bind. But maybe we should think about the DNA that's wrapped up in chromatin. And DNA that's wrapped up in chromatin, that is wound around a histone core. And the histone core is shown here, and DNA is wrapped around it with about 2 turns. But when NFkB wants to bind the DNA, it looks like this. Here's the DNA in cross-section. NFkB really hugs the DNA, right? It embraces it. So that these two structures are not compatible with each other. If NFkB wants to bind the DNA, it has to kick off the histone core. So here's the histone core with DNA wrapped around it. And when NFkB comes, it has to unwrap it until it kicks off the histone core. So how would that work? Well, it works basically by disrupting the contacts of the DNA with the histone core. And there are 14 contact points. That's known from biophysics. So the 14 contact points are shown here as an enhancer that is in a very deep sleep, in a completely closed latent enhancer. And when it can be opened until it is fully opened and active as an enhancer in the E0 state. And each of these 14 transitions is driven by the transcription factor, NFkB, through this Hill function. So we can test this model by feeding our signaling data that I showed you earlier into the model and predicting the enhancer state. What fraction of the DNA is in an open configuration? You see, in response to TNF, very little is opened. But in response to PAMP-CSK, CpG, and LPS, many cells will open their DNA. And in response to poly-IC, very little. So that's a prediction. That prediction was tested experimentally using A3K4 monomethylation ChIP-seq, which is an enhancer mark, right? And we actually get 2 clusters here. One cluster is interferon, associated with interferon genes, and that's different. But one cluster is associated with NFkB. And you see that these NFkB enhancers are induced highly by LPS and by CpG and PAMP-CSK, but not by TNF and poly-IC, just like what we predicted. And what's the difference between them? Of course, it's the oscillations. This one oscillates, these ones do not. And so we tested this further by using the mutant that doesn't oscillate in response to TNF. And indeed, that mutant leads to opening of chromatin, even in response to TNF, much more so than the wild-type counterparts. And after opening the DNA and creating new enhancers, then these cells are no longer the same kind as their healthy counterparts. They're different in their chromatin state, and therefore they respond to a secondary stimulus quite differently. And that's shown here, that these are inducing genes now that are otherwise not inducible with the secondary stimulus. So in other words, we learned something about how oscillations play a role. They play a role in creating new enhancers. And that when cells are oscillating, when transcription factors are oscillating, they are not affecting the epigenome. But when they're not oscillating, they can reprogram the epigenome. So in other words, TNF-induced oscillations allow for gene activation, but without triggering epigenome remodeling, without creating memory, right? And so that was an exciting insight. So with that, let me thank the people who did the work. I think I've already mentioned all started with Brooks and Ade. And then subsequently, the last piece of study was done by Quen Cheng. Stephanie Lücke played a role. Apeksha Singh had another study. I also talked about the modeling work from Shalu Guo, and many other people in the group contributed to other aspects of the work. Thank you very much for your attention. Of course, I'm happy to answer questions.
They are just amazing, Alex. In fact, we are waiting for the question, but before making my question, I would like to comment that in the beginning, you show all the complexity, and then you start to show to us how modeling, how mathematics can help us to solve or to help us to understand all this complexity.
That's right. Yeah, it's amazing. There's some actually simple principles that emerge, and those principles actually make sense, right? They're not so difficult to understand. But it only makes sense after you've done a lot of work and you have to get through all of these difficult things. Then in the end, everything makes sense.
Yeah, I have some question here that I will read, but there is because before going to your lab 2 years ago, I looked at one interview from you where you said that in the past century, physics start to use mathematics to, and it produced the revolution in physics that now we are enjoy here with this online meeting. Then you propose that on this century, if biology start to use mathematics the same way, then we will have a real revolution that will result in better life for people.
Yeah, I think we're in the middle of that revolution. Of course, we now have we have another kind of mathematics available, not just deterministic mathematics. We have stochastic mathematics, and then we have data science mathematics. And so the ability to model is there's been a lot of innovation since the revolution that took physics. And so we have more tools at our disposal. Yeah.
Yeah, absolutely. I have here one question from Elnard Brunet, that he is one of the organizers of this
Hmm.
talk. And his question is, when cells make a consortia to decide, how should they communicate with each other?
Great question. Great question. In fact, you put your finger on um, one of the questions that we have not answered is we don't know how they communicate. And maybe they don't. This is actually more a thought experiment this time. So a whether macrophages really leverage each other's information is not clear to us at this time. So So good question, but not one I can describe in mechanistic detail. Yeah.
Great. You showed that some cells behave correctly and some others do not behave correctly, but it seems to be just fine. My question is, maybe is there any advantage of some cells behave not correctly on this context? Maybe they will do a good job in another context, or this is just a variation that is just fine?
Yeah, I think that's a really interesting thought. So in many systems, you are right that there is, in fact, an advantage to variability, right? And that may very well be true in the case of the macrophages too. On the other hand, macrophages, as I mentioned in the introduction, they really are working as individuals, maybe as small consortia, like I mentioned. But there's no organ of macrophages, right? They are distributed as single cells everywhere. So that's why our hypothesis has been that specificity and the reliability is an important characteristic. But it's possible that variability, some variability or uncertainty or sloppiness might also confer some advantage, but I don't know what that might be. A good question to address in the future.
Okay. We have more question here. Is from Aline Oliveira Lima. She is asking, is it possible to identify immunogenetic profiles that can predict exaggerated response to some type of infection or even vaccination?
Yeah, that's a great question to make this work also more um, valuable for biomedical research. So what we're trying to do, we know that some macrophages are better at their job than others. But in clinical studies, we can't really we cannot assay macrophages for their functions. We can only assay macrophages for their RNA or their cell surface markers. We can only get a snapshot. So what we're attempting to do now is to develop molecular markers, gene expression signatures of macrophages that are working that healthily, have a good immune response specificity versus those that have a poor immune response specificity. And when we have those molecular markers, then we can interrogate them. We can use them to interrogate clinical data sets. And including spatial transcriptomic data or things like that, to be able to figure out when we identify macrophages in these clinical data sets, are they functioning healthily or are they functioning unhealthily? And that would give us a way of relating these macrophage functions that I described to disease and health in people. So anyway, that's the future. It's a great question. So maybe you want to join us in answering it.
Uh, I think that we have time for one question more. Okay, there is one question here. I prefer to ask the question from Mariana Teixeira de Freitas. She is my student and she also have good questions. It's a
Great.
long question. Try to in Portuguese. I will try to. Um, she's asking if there are specific mechanisms that allows the microphage to compensate the failure of the others.
Ah, yeah, that was a good question. Actually, that's a great question. And I think it's similar to the question that your co-organizer asked. How are macrophages in a consortium working together? And actually, we don't really know yet. At this point, it was just a thought experiment. And but given that the thought experiment tells us there is an advantage to work together, I would suggest that there probably are ways that they can work together, and so we should find out the mechanisms. We know that macrophages signal to each other through TNF and other cytokines, but whether this is really the mechanism by which they can work together, we don't know. So, but it's a great question. Yeah, we should look into it.
I told you she has good question for Quentin. Oh, I have one more. I mean, look at the time, as we have time for other question. Let's see. Victor Gonçalves Rodrigues is asking you, considering that NF-κB works like a central regulator of immune response, how your research can help to develop therapies that are more directed to specific case.
Yes, I think great question. So it is a central regulator of the immune response. And of course, pharmaceutical companies have spent a lot of money trying to develop drugs to inhibit NFκB. But because it is so central, when you inhibit NFκB, you may alleviate one symptom, but you cause a lot of other problems. So the side effects are really enormous. And that's why there are no good NFκB inhibitors in the clinic. There are many good NFκB inhibitors in chemical companies that we can use for our research, but none of them working in the clinic. So I think what this work suggests is that we have to target not NFκB, but we have to target specific dynamic features. For example, non-oscillations. If we can convert non-oscillations back to oscillations, that will still allow NFκB to function and activate gene expression, but it will remove the epigenetic remodeling. So we explored this idea in some papers where we described that our pharmacological goal should be to target not a signaling protein like NFκB, but our pharmacological goal should be to target the dynamic feature of a signaling activity. And so how do you do that? Well, you do sensitivity analysis and you can do high throughput computation with these mathematical models to identify reactions that are specific for each dynamical feature. And but those drugs are not yet in the clinic. This is all just proof of concept studies. So, we still have some way to go. But that would be the strategy to improve immune regulators, or modulators of immune responses that are specific and don't have these sort of side effects that we currently have.
Yeah, this is like if NF-κB is not working well, do not kill him, just make him work better.
Yes, exactly.
I think we have maybe time for one question more. This question comes from Arnaldo do Nascimento. No, this question comes from Maria Eduarda. She's also my master degree student.
Great.
Yeah, I have a lot of questions for my lab.
Thank you.
Oh, okay, she said, good evening. In genetically identical macrophages, what determines that some cells exhibit NFkB fluctuation while others do not? Does this heterogeneity arise from variations in the level of IKK and the regulatory proteins?
Yeah, great. That's a great question. And in fact, it's a question I think I can answer because that is one of the nice things about the mathematical model, right? Because we can fit the model to the data and thereby determine the distribution of parameters that are required to capture the phenotypic heterogeneity that we observe in the experiments. And so what we find is that one of the key points is the receptor expression level. So the receptor expression level is different in different cells, and we can infer the distribution, and then we can measure that distribution. And that seems to be a key point. A second key point actually is after receptor and ligand are binding, they are usually internalized into the cell. And the lifetime of that is also important, how quickly it is degraded. And that varies between different cells. And IKK abundance can vary, but it doesn't play a big role in the heterogeneity. So it is not very important. There are some other activities that do play a role in the heterogeneity, but I think those two are particularly important. It's the receptor abundance and the lifetime of the receptor signaling complex. And so those are the main ones.
This is very interesting because Maria Eduarda is building a new model about initiation of breast cancer, maybe next year, if we are founded for the next CISBio Rio, maybe you will come here and you have the opportunity to talk directly to her.
Oh, that would be fantastic. Yeah.
I have just one question more, and he just to finish. Sorry for so many questions.
Oh, I love it. Yeah.
I know. This question comes from Arnaldo Levino do Nascimento. His question is, of the six signaling codons you identified, which do you believe is easiest to manipulate with a drug to control uncontrolled inflammation? For example, is it easier to stop the oscillation or reduce the overall activity?
Yeah, I think that's a great question. You know, the simplest the existing drugs, I should say, usually try to inhibit IKK. So what that does, it of course reduces the peak activation. But because it reduces the peak activation, it also reduces the negative feedback. And as a result, you reduce the peak activation and you prolong the response. And you may also not just prolong the response, you may convert oscillations into non-oscillations. And so actually both of those are bad because both of them lead to chronic inflammation. So it's not a surprise that the available drugs that target IKK, the initiator, if you reduce that by 90%, great, you reduce the amplitude by maybe by 90%, but you have created a problem in generating, you created a chronic inflammation problem because the negative mechanisms are now too weak. But I think there's some other drugs that can, if you're just looking at various compounds that affect the signaling network, there are many different other things you can manipulate, but usually you need a very precise dose. That's one of the problems with it. The dosing has to be precise to get a precise effect. And of course, that is very difficult to achieve in the clinic. So I don't have a really good example yet for a drug that's very promising, unfortunately. Maybe in the future.
Great. Alex, thank you very much. We learned a lot, and we appreciated very much your talk and all the conversations. Thank you to everyone that make all these great questions.
Yeah, thank you so much for your interest and the great questions. And I look forward to being in touch with you. Feel free to send me any follow-up emails if you like. Thank you.
Absolutely. Thank you. Bye-bye.
Bye-bye.
Alex, você tá aí? Alex brasileiro?